Welcome, biology student, to lecture 7-4, the diversity of cellular life. This is Mr. Worley here, and I'm glad you're with me. Before we start, I want you to make sure that you have a piece of paper handy so that you can take a full page of notes and, after that, do your section assessment. I also want you to make sure that you have your book with you so you can follow along as you read. Class, let's look at our learning objectives. We want to describe cell specialization, and we want to identify the organization levels in multicellular organisms. Your lecture exercises are on page 193, numbers 1 through 4. So, without further ado, let's start reading. Here we go. It says that life is cellular. Look closely at a part of living thing, and what do you see? Hold a blade of grass up against the light, and you see tiny lines running the length of the blade. Examine the tip of your finger, and you see the ridges and valleys that make up fingerprints. Place an insect under a microscope, and you see the intricate structures of its wings and the spikes and bristles that protect its body. As interesting as these close-up views may be, however, they're only the beginning of the story. Look closer and deeper with a more powerful microscope, and you'll see that there is a common structure that makes up every living thing, the cell. Seeing is believing, an old adage saying goes. It would be hard to find a better example of this than the discovery of the cell. Without the instrument to make them visible, cells remained out of sight and therefore out of mind for most of human history. All of this changed when a dramatic advance in technology, the invention of the microscope. Early microscopes. It was not until the mid-16th century that scientists began to use microscopes to observe living things. In 1665, Englishman Robert Hooke, I want you to know that, Robert Hooke might be a good quiz question, used an early compound microscope to look at a thin slice of cork, a plant material. Under the microscope, cork seemed to be made of thousands of tiny empty chambers. Hooke called these chambers cells, because they reminded him of a monastery's tiny rooms, which were called cells. One of Hooke's illustrations of cells is shown in figure 7.1. The term cell is used in biology to this day. We now know, however, that cells are not empty but contain living matter. In Holland, around the same time, Anton van Leeuwenhoek, used a single lens microscope to observe pond water and other things. To his amazement, the microscope revealed a fantastic world of tiny, living organisms that seemed to be everywhere, even in the very water he and his neighbors drank. Blah. Very cool. Before we move on to the next page, let's review that we have some vocabulary: cell, cell theory, nucleus, eukaryote, and prokaryote. Our key concepts is what is cell theory and what are the characteristics of prokaryotes and eukaryotes. Let's move on to the next page, 170. Page 170. The cell theory. Soon, numerous observations made it clear that cells were the basic units of life. In 1838, German botanist Matthias Schlechten concluded that all plants were made of cells like the one in figure 7.2. The next year, German biologist Theodor Schwann stated that all animals were made of cells. In 1855, the German physician Rudolf Virchow concluded that new cells could be produced only from the divisions of existing cells. These discoveries, confirmed by other biologists, are summarized in the cell theory. Nice. Vocab word, should know it. A fundamental concept of biology. The cell theory states, all living things are composed of cells. Cells are the basic unit of structure and function in living things, and new cells are produced from existing cells. Oh, man, this looks like a good quiz question. What do you think? Exploring cells. Exploring the cell. Following the footsteps of Hooke, Virchow, and others, the modern biologists still use microscopes to explore the cell. However, today's researchers use microscopes and techniques more powerful than the pioneers of biology could have imagined. Researchers can use fluorescent labels and light microscopy to follow molecules moving through a cell. Confocal light microscopy I'll tell you what, if you give me a half-page research paper that you typed, I'll give you extra credit points for that. What is confocal light microscopy? Which scans cells with a laser beam makes it possible to build three-dimensional images of cells and their parts. High-resolution video technology makes it easy to produce movies of cells as they grow, divide, and develop. Wow. Let's look at this little graphic here. In 1665, Robert Hooke made the first microscope. Later, Van Leeuwenhoek observed tiny living organisms in water. Let's move on to page 171. Moving to page 171. All right. These new technologies make it possible for researchers to study the structure and movement of living cells in great detail. Unfortunately, light itself limits the detail of resolution of images that can be made with the light microscope. Like all forms of radiation, light waves are diffracted or scattered as they pass through matter, making it impossible to visualize tiny structures such as proteins and viruses with light microscopy. By contrast, electron microscopes are capable of revealing details as much as 1,000 times smaller than those visible in light microscopes, because the wavelength of electrons are much shorter than those of light. Oh, yeah, this looks like a great quiz question. Why do electron microscopes make much closer images than light ones? Transmission electron microscopes, TEMs, make it possible to explore cell structures and large protein molecules. Because beams of electrons can only pass through thin samples, cells and tissues must be cut into ultra-thin slices before they can be examined under a microscope. With scanning electron microscopes, SEMs, a pencil-like beam of electrons is scanned over the surface of a specimen. For SEMs images, specimens do not have to be cut into thin slices to be visualized. The scanning electron microscope produces stunning three-dimensional images of cells. Because electrons are easily scattered by molecules in the air, samples examined in both types of electron microscopes must be placed in a vacuum in order to be studied. As a result, searchers chemically preserve their samples first and then carefully remove all the water before placing them in a microscope. This means that electron microscopy can be used to visualize only non-living preserved cell tissues. And let's continue to look at our neat little timeline. After Van Leeuwenhoek, I can't say his name, Matthias Slijden concluded that all plants are made up of cells. Then Virchow discovered that all cells can only come from other cells. And finally, back in Schwann said that all animals are made up of cells. And finally, Linus Margulis proposed the idea that certain organelles, tiny structures within some cells, were once free living cells themselves. I'm going to put a big question mark by that. That is more of a faith statement than a science knowledge statement. Anyway, before we continue on, I'm just going to take a little bit of a pause and say that, class, I hope you are taking notes. I hope that you are writing down the main subsections, you should be writing down the vocabulary, the points of the cell theory, and perhaps this little timeline. How was the cell theory developed and who did it? All right, let's rock on to the next page, page 172. Okay, page 172. Go ahead and read along with me. Class, we're going to move on actually, class, I'd like you to please go to the next video. Go to section 2, video for section 2,